Thank you, Julie. It's my pleasure to be able to introduce today's speaker, Dr. Diana Taft. I'm particularly pleased to do that because we share, in some ways, a common origin, i.e., we both grew up in the state of Ohio, although in different generations, I would say.
Ohio was grad school. Grew up is complicated.
Maybe pull the mic just a little closer.
Oh. Is that better?
So anyway, Dr. Taft received her PhD from the University of Cincinnati, and then she moved on to do postdoctoral work at Davis in California, and she again came here as an assistant professor in, I guess it's food and human nutrition, something like that. That's the department at IFAS that she's a member of, and she's interested, and we share another crossover point, and that is I had some background in pediatrics, and her topic is the infant microbiome, how important it is, where does it come from, and maybe even what it does to the infants. But anyway, she's going to tell us all about that. So welcome, Dr. Taft.
Thank you. Could you bring my... All right. I am going to tell a story today that I think is one of the most interesting I've encountered so far. And it is the story of a microbe that is vanishing. And it's a microbe that I'm going to argue is really important to health. So what's happening to bifidobacteria in infants? Better? Okay. Can do. All right. Too much? Okay. All right. So at this point, you've all heard at least a little bit about the gut microbiome, and this is very much something that started to explode as a topic of research interest with the invention of next-generation sequencing, because all of a sudden we were freed from having to culture every organism to figure out what it was, to be able to just take all the DNA, throw it on a sequencing platform, and start to answer our questions. And everywhere we looked, we started to see the importance of these microbes. Asthma, allergy, diabetes, cancer, everywhere we looked, it was a whole new organ system that we had just neglected. And it all begins in infancy, because barring some kind of infectious process affecting the placenta, we don't really acquire live bacteria until we're born. And it starts to assemble rapidly during those first 2 years of life and has the potential to influence our health all throughout our lives. So what does the infant microbiome look like today? Well, today it depends a little bit on whether or not an infant is born vaginally or by C-section. So in the case of a vaginal delivery, you see a lot of Bacteroides. In the case of a C-section, you see a lot of Firmicutes. And every now and then, you see just a little bit of this green here that is sorry, they just changed the phylum name on me. I'm going to go with the old name, Actinobacteria, which is the phylum to which genus Bifidobacterium belongs. Wrong button. But here's the thing. We know something about what the infant gut microbiome looked like more than a century ago, because right at the turn of the 19th into the 20th century, people were fascinated by microbiology, and what they were doing is they were taking fecal smears, putting them on a microscope, and describing what they saw. So we know 1906, 1913, that if you did that from the feces of a breastfed infant, what you would see would be a near monoculture of what was then called Bacterium bifidus, in which today we would call various species of Bifidobacterium. So wait a minute. I just showed you the slide that goes today that said very, very small amount, only if it's a vaginally delivered infant. But 120 years ago, if we did the same thing, it would have been a near monoculture. What happened? Well, we have some hints, and the hints start in human milk. And human milk is this incredibly complicated, absolutely fascinating biofluid. And if you look at human milk, it's mostly water. All right, forget about the water. Not that interesting. But once you do that, the third most abundant component are these things called human milk oligosaccharides. And these are these really specialized sugars. And they're interesting because they're not digested by the human infant. So you put in human milk oligosaccharides in one end, and if there were no bacteria or other microbes, they would come out the other end unchanged. Third most abundant component outside of water in human milk. That is a massive investment of energy. Why are moms making these if the baby's not digesting them? Well, what's really interesting is they're for the microbes. So they play a couple of different roles. If you look at them, there's one particular species of Bifidobacterium with the very long name, Bifidobacterium longum subspecies infantis. It ain't never met an HMO it doesn't look at and go, showtime! We also have some species of Bacteroides that can consume these. Some other species of Bifidobacteria will consume some of these, and they can act as decoy receptors for pathogens to prevent an infant from becoming infected. So they have a bunch of different roles. And it's really interesting what happens when you have HMOs and a bifidobacterium that can eat them, because what you get out the other end of the bifidobacteria is just a ton of short-chain fatty acids. Massive, massive amount of acetate and lactate, to the point that you will dramatically lower the pH of the infant feces. And as we start to look at these short-chain fatty acids, we start to see more and more roles for them within human health. So they show up as really important to intestinal homeostasis. So they're important for how the intestine develops and how it maintains its function. They're important to T cell and immune regulation, so how the immune system develops and functions. And they're a really important energy source for enterocytes. So some of the main cells in the colon depend on these short-chain fatty acids for energy. So they're really important to how the intestine functions. Well, this system now makes a little more sense. Moms invest massive amounts of energy. The bifidobacteria converts it into a form that promotes infant health. Aha! So what's going on here? And this paper was sort of my first hint and the first moment where I really started to understand what I think might be going on with bifidobacteria. And this is a truly brilliant concept by Justin Sonnenberg out at Stanford, where what he did was this very, very elegant mouse experiment. He took mice, he fed them a standard chow diet with lots of fiber, and he saw what level of microbial diversity was present. He then switched them to a mouse diet with very low levels of fiber. That fiber is what feeds the microbes. You can call it microbially accessible carbohydrate. So that would be the carbohydrate that the host does not digest. So the mouse doesn't digest it, the human doesn't digest it. So it reaches the bacteria within the intestine. And what Sonnenberg did is he high in max, low in max, and then did the next generation and put them back on that high max diet. And all the microbes that had disappeared during that period of low MAC, they were extinct, they were gone. Because they were no longer part of the population, they could not come back. So even after they went back to that high MAC diet, the microbes that had died out from starving for lack of these carbohydrates couldn't come back. They were gone from the population. And we can sort of start to look at the genetic capabilities of some of these different bacteria. So I've already mentioned Bifidobacterium longum, subspecies Infantis. This is the one that never met an HMO it doesn't like. It's got a really closely related subspecies called Bifidobacterium longum, subspecies longum. That one never met a plant oligosaccharide it didn't like. So it's got all this genetic machinery that it needs to consume the fiber in plants, but very little of the genetic machinery it needs to consume the human milk oligosaccharides. B. infantis, despite being very genetically similar to B. longum longum, has all the genetic machinery it needs to consume all the HMOs, and very little of the genetic machinery it needs to consume the plant fiber. Okay, so I'm starting to get a picture here. And indeed, I keep hitting the wrong button. And indeed, you sort of look and go, well, what were people feeding their kids 100 years ago? And it was really a very simple choice. You figured out how to breastfeed that kid, or that kid had a very good chance of dying, because the burden of infection pathogens 100 years ago was that high. So the selective pressure was everybody, if you could figure out how to do it, you breastfed, because formula was like cutting edge technology, and it was really only for the most dire emergencies. So the idea of having a formula-fed infant, oh, that was rare. And that meant almost everybody was on a high, all these babies, they're on a high MAC diet. They're on the breast milk diet that has all these human milk oligosaccharides. These are not things found in formula. So what have we done to our kids? As we switched from breast milk to formula, we did what Sonnenberg did to his mice. We switched from a high MAC diet to a low MAC diet. What are going to be the microbial consequences of that? And sure enough, if we look at formula-fed infants, the rare ones who were around 100 years ago, what do we see? We see a lot of Lactobacillus acidophilus, some coliform bacteria, some enterococci. All the things we see, even in the breastfed infants, today. Who survived? Who we fed? And indeed, you can sort of track breastfeeding rates in the US. And in many ways, it was very tragic, because if you look at the nadir in breastfeeding in 1973, less than 1 in 4 infants received a single feeding of breast milk in the hospital before they went home. Almost everybody went straight onto formula. And then you can sort of track, at the same time you see these drops in breastfeeding rates, you see an increase in fecal pH. That's that loss of the short-chain fatty acids. If you're not taking those human milk oligosaccharides, converting them to the short-chain fatty acids, you wind up with a higher infant fecal pH. So you're starting to change the whole environment of the gut. Indeed, that Gerald Tannick, who's probably one of the greatest microbiologists around, and probably the person who knows more about Bifidobacterium at all, has this lovely quote: B. longum subspecies Infantis is probably the most intensively studied of the Bifidobacteria with respect to biochemistry, but it may be an endangered species in Western countries. Uh-oh. Well, that leaves a very simple question. Do we care? Does it actually matter to infant health? All right, let's walk through a few studies and look at some of the ways that bifidobacterium matter for health. With this crowd, I'm hoping I don't have to sell too hard the idea that vaccination saves lives. And this is old data, so pre-COVID, so the number's probably higher now, but it saves at least 6 million lives per year. But to be effective, vaccinated individuals must mount a robust immune response to the vaccine. And bifidobacteria for infants are part of that story. So this data comes out of a Bangladeshi cohort where we collected stool samples at week 6, 11, 15, and age 2 years, and really dug deep to understand not just sort of the genus level differences in the gut microbiome, but some of the species level differences. And this is interesting because this is Bangladesh. So everywhere you see blue, that's B. longum subspecies infantis. Is not endangered in this cohort. And we can look at this, and this was part of Nazmul Huda's doctoral dissertation. I was a co-author on the paper. But what Nazmul figured out is that, yes, bifidobacterium at levels at time of vaccination are correlated with how robust your immune response is, both at the time and 2 years later. So if your baby has very high levels of bifidobacterium and you give them a vaccine, they're going to respond to it at a higher level and have a more durable response than if the baby does not have high levels of bifidobacteria. But that this isn't something that happens consistently for all species of bifidobacteria. To highlight my favorite, B. longum subspecies infantis, that's very much the positive association with that vaccine response. But the closely related B. longum subspecies longum, no benefit in terms of vaccine response. So it's not just enough to have high levels of bifidobacteria. You need to have the high levels of the right bifidobacteria to see a benefit. So, conclusions from this part. Yes, we probably should care about bifidobacterium because high levels of bifidobacteria in early life correlate with improved vaccine response in early life, and that improved response is sustained 2 years on. And we can't just say any bifidobacteria, it's got to be the right bifidobacteria because not all subspecies and species have the same effects. All right, so this is one thing. But if you're living somewhere not like Bangladesh, where the burden of infectious diseases are lower, should we still care about bifidobacterium? Well, this brings in some of my work as a postdoctoral research fellow at UC Davis, where what we wound up looking at is antimicrobial resistance. And this is a huge problem worldwide for everybody because we've got a minimum of 2 million illnesses per year in the US that are caused by antimicrobial resistant pathogens and a minimum of 23,000 deaths per year. These are deaths that in the absence of resistance would be preventable. So having these resistance genes in circulation within our gut commensals invites them to spread to other places. And in fact, this has gotten to be such an issue that even infants as young as 1 week of age can harbor massive high levels of antimicrobial resistance genes. So why would antimicrobial resistance be a problem if it's in a commensal or a beneficial bacteria? Well, let's say that baby, I don't know, picks up strep throat. So now that baby needs to be on antibiotics to treat the strep infection. But those antibiotics are also going to influence the gut microbes. And it's going to select for two things. It's going to select for bacteria that already have the resistance gene, and it's going to put pressure on the bacteria that have the resistance gene. If it's a transposon or sort of another form of transferable resistance, it's got pressure to give it to the other microbes. And all of a sudden, you can be in this situation where you've totally shifted the gut composition and you've enriched it for these antimicrobial resistance genes. And as you have more and more, the chance of them transferring to the next pathogen that baby gets sick with starts to go up. So way better to just not have it in the first place than to risk it transferring to a pathogen later. And the good news is, bifidobacterium, because of their genetics, they don't like to go acquire a lot of antimicrobial resistance genes. They don't like to acquire a lot of genes from other bacteria at all, which on the one hand drives microbiologists nuts because they're very hard to study genetically. On the other hand, as our gut commensals, that's good news for us because they're not particularly likely to carry a lot of these resistance genes. And so this sort of led us to this hypothesis that you've got a baby with very high levels of bifidobacterium, and they're probably not going to carry a lot of antimicrobial resistance genes because all that acid the bifidobacteria produces tends to kill off a lot of the E. coli and some of the other species that are more prone to carrying resistance genes. So we went to that same Bangladeshi cohort I talked about, and we went looking at the different levels of bifidobacteria in early life. And the bulk of these kids had very high levels of bifidobacteria. So more than 50% of their microbiome was bifidobacteria. But there were a handful who had lower levels. And this was back when whole metagenomic sequencing was still expensive. So we went ahead and selected all the infants with less than 20% bifidobacteria and a handful matched on age and gender with high levels of bifidobacteria. And we sent them off for whole metagenomic sequencing. We also found a cohort of infants from Sweden. All 100 of them had had whole metagenomic sequencing done. So we went ahead and used the same definitions of high and low bifidobacteria that we used for the Bangladeshi infants, and would repeat the same analyses in a different cohort. What did we find? Sure enough, having those high levels of bifidobacteria in early life meant infants had reduced carriage of antimicrobial resistance genes and a reduced diversity, so fewer different kinds of antimicrobial resistance genes within their gut microbiome. We got super excited because it was like, oh God, it's going to be like the vaccine story. Have we just totally changed the trajectory? No. Because what happened was actually quite interesting, where while the infants have the high levels of bifidobacteria, that's that reduced diversity of antimicrobial resistance genes. But as you introduce complementary foods, you start to shift the gut microbiome, and those infants acquire a greater diversity of antimicrobial resistance genes. At the same time, the low bifidobacteria infants start to acquire a greater diversity of other microbes that dilute the absolute level of the resistance genes. So over time, you have the high bifidobacteria infants that come up and have a greater diversity of antimicrobial resistance genes. And you have the low bifidobacteria infants come down and have a lower abundance of resistance genes. Sweden. different profile from Bangladesh, where here the majority of the infants met that low category, and only a handful met the criteria for high bifidobacteria. So a little bit different from what we saw in Bangladesh. The BIFs are rarer, and when they're present, they're not quite the same high level you saw in Bangladesh. But you saw that same effect, where if they did happen to have high levels of bifidobacteria, they did have that reduced abundance and reduced diversity of the antimicrobial resistance. And the low infants sort of came down in that abundance over time. So in conclusion, high levels of bifidobacterium are associated with reduced abundance and diversity of antimicrobial resistance genes in early life. So these kids, if they're lucky enough to have high levels of bifidobacteria, they're less likely to have a problem with antimicrobial resistance. And infants with low levels of bifidobacteria have a drop in resistance gene carriage during weaning, while the diversity increases in infants with high levels of bifidobacteria during weaning. So everybody winds up in about the same place. They just take very different routes to get there. And we really need more studies to understand the timing of the sort of bifidobacterium effect. And there are a handful of kids in that high bifidobacteria group who just never seem to come up. I would really like to know what those kids are eating, because I'd like to feed that to more kids. So future studies. All right. Here's a question, and this is one that started to haunt me. If B. langum subspecies infantis is endangered in the Western world, what on earth were the bifidobacteria in those Swedish kids? And what does that mean in terms of their health? Well, I was very fortunate because with much scrounging around, I got access to a bunch of different cohorts where we were able to go through and identify not only the sort of absolute level of bifidobacterium as a genus, but the different species and subspecies present in each sample. And for this work, we targeted infants who are about 2 months of age at the time of sample collection. So we had 2 US cohorts, one from California and one from Ohio. We had a cohort from the Gambia, the cohort from Bangladesh, and then we had 4 European cohorts from Switzerland, Germany, Finland, and Austria. If you look at breastfeeding today in these cohorts, rates were very high. Ohio was the worst with only 80%, but still better than less than 1 in 4. So the breastfeeding has come back. Go moms. But you see differences still in duration, where if you look at the Gambia and Bangladesh, the average duration of breastfeeding in both of these countries, more than 2 years. Somebody's listening to the World Health Organization because they recommend for 2 or more years. And actually, Bangladesh is really funny because when the World Health Organization came out with that recommendation, Bangladesh saw a drop in breastfeeding duration. And their public health people had to go out and run this whole campaign of, don't listen to the World Health Organization. You can breastfeed longer. You don't have to stop at 2 years. So huge, huge investment of time by mothers to breastfeed these kids. Whereas if you look in the US, we get 6 months, but we don't really get a year. If you look in the European countries, you get 6, 8 months. So much shorter duration. And on top of that, you can sort of dig into the history of breastfeeding in these countries. Bangladesh and the Gambia, they've always had that 2 or more years of breastfeeding. They've never had widespread adoption of formula because 1, people couldn't afford it, and 2, when the formula companies tried to move in, to those regions, there were massive boycotts and protests, and they never really got established in the same way. In Europe, it's interesting because they never had that same very low rate of initiation. They always had mothers breastfeed, but it was for very, very short durations of maybe only a few weeks. And then, of course, there was the mess in the US where we just didn't breastfeed at all, period, full stop. So what do the infant gut microbiomes look like in these countries today? Everything black is something that's not bifidobacteria. Everything colored is a bifidobacteria. Let me make a point. These are all exclusively breastfed infants. So these are all infants who should have been in that high bifidobacteria category. No bifidobacteria at all of any species. No bifidobacteria at all of any species. No biff, no biff, no biff, no biff. Teeny tiny little bit of biff. Poof. Vanished. And it's not just in Davis. In Cincinnati, those exclusively breastfed infants, nice little chunk of them with vanished bifidobacteria. And we can also look at it and we can go, what are the species? Well, we have a couple of infants in Davis who had B. longum subspecies infantis. And I have an anecdote about those babies I'll tell at the end. And then we see a lot of B. longum longum, a lot of B. breve. So we don't see the bifidobacteria that are associated with that improved vaccine response. We've changed how the immune system functions in these kids. Cincinnati, what do we see? Longum longum, breve, a little bit of bifidum. We've changed, again, what they see. All right. Something a lot happier. Gambia and Bangladesh, these solid walls of blue. So the countries where you didn't move away from breastfeeding, you still see those sky-high levels of B. longum subspecies infantis. What about the European countries? So these never had that same dip in initiation, though they did have a dip in duration. And they came back to longer duration breastfeeding in Europe much faster than we did in the US. We see bifidobacteria, but you sort of have to hunt and peck to find the B. infantis. Some countries, it's a little bit rarer than in others, but all of them have at least a little bit, but it's mostly B. longum, B. longum pseudocatenolatinum, or sorry, B. pseudocatenolatinum, B. bifidum, B. breve. You just don't see that B. longum subspecies infantis. And quick comment, because when we talk about the infant gut microbiome, everybody talks about vertical transmission from mom. What survives in the gut is what you got from your mom. These studies were all done in Europe, in the US, in other countries with this disrupted breastfeeding pattern. But when we look at sort of this B. longum subspecies infantis, we start to see a little bit of a different story. Because in Davis, we had a really interesting thing occur. We tested those same infants at 1 week of age, at 1 month of age. No B. longum subspecies infantis. All of a sudden, at 2 months of age, we have 3 kids with B. longum subspecies infantis. Here's my anecdote. At the same time the samples for this study were collected, there was another study running in Davis. It was a probiotic trial looking at B. longum subspecies infantis as a probiotic in healthy term infants. The 3 kids who picked up the B. longum subspecies infantis, I know this because the clinical research coordinator enrolled for both studies. They went to the same daycare as a kid who got the active probiotic in the other trial. They picked it up at daycare. That's not vertical transmission from mom to baby. That's horizontal transmission from baby to baby. In Bangladesh, where you have that sky-high level of B. infantis, and almost all the babies have at least a little bit of B. infantis, it begins to disappear from the microbiome by age 2, where as babies are no longer breastfed, they lose the B. longum subspecies infantis. I think what we're looking at here is a disrupted infant gut microbiome where we ran Sonnenberg's mouse experiment in our babies. We took them from a high MAC diet, we put them on a low MAC diet, we've taken them back to breastfeeding, and the microbes have not come back with it. So, what can we conclude? Patterns of bifidobacteria colonization can be grouped by country history of breastfeeding. If you happen to live somewhere where all the babies have been breastfed and have been breastfed for generations, you probably have B. longum subspecies infantis. If you don't, you probably don't. So the question then, is this species got a different epidemiology? Does it depend on infant-to-infant transmission? And if it does, and if we think this is important to infant health, and I'm convinced it's important, what can we do to bring it back? Well, I've got a study starting, and I'm really hoping some of you might have new grandbabies or a grandbaby on the way, because what I'm trying to do with this study is run lactation support groups for mothers and babies. We'll have an IBCLC, so an international board certified lactation consultant, there to run the meeting. We'll give half the babies an L. reuteri probiotic. We give the other half B. infantis. And can these lactation support groups become a point of transmission of a beneficial commensal for infants in the US? Can we bring it back? So if you happen to know someone who's having a baby, or you're going to have a grandbaby in Alachua County soon, I have study flyers, and I'm hoping you'll pass it along. All right, thank you for your time, and I'm more than happy to take any questions.
Questions? Couldn't have been that crystal clear. Joe? As a well-known infectious diseases person?
None at all. Oh dear.
No comments?
Well, what I would like to know is if you take a pure culture of the bifidobacterium and put it in somebody else that's already doesn't have the bifidobacterium, what happens to it?
So, been done, because that was that probiotic trial I mentioned, and it depends a little on the age, where if it's a young baby and you give it for a couple of weeks, it's going to stick around until mom stops breastfeeding.
Now, what happens in, you haven't done anything in adults?
No, this is not, this is
This is all
in
all in infants, right?
Yep.
Okay.
Yeah, if you're not eating a human milk oligosaccharide, it doesn't have anything to stick around.
So you said that depending on the microbiome, there's a greater or lesser immune response to vaccination.
Mm-hmm.
Question is, if you look at the other end at disease, is there a correlation or a reverse correlation between the amount of the bifidobacteria?
So I can't answer directly for the bifidobacteria. but I can say fecal pH, the lower your fecal pH, the higher your levels of bifidobacteria. And a low fecal pH in Gambia is highly predictive of protective. So the lower your fecal pH, the more protected you are against all causes of morbidity if you are a Gambian infant. And we know it's all B infantis in Gambia, and we know that correlates, so.
I think one of the questions is that we know in viruses we have viral interference. So if you already have certain viruses in your GI tract, others can't really do well. Question is, in kids that have a healthy microbiome, does that prevent bacteria and enteric bacteria that might be negative from colonizing the gut?
Oh, yes. And we think that's actually part of the fecal pH story, because a lot of the pathogens are not particularly acid tolerant. So you can get a fair amount of protection against a lot of GI illness just by dropping that pH. But yes, there is totally a gut microbiome role for colonization resistance, and I'm trying to get a study funded to look at that too. Waiting to hear.
I think one of the interesting things that's happened, actually not so long ago in adult medicine, and probably in kids as well, is the notion of fecal transplants in kids that, or people who get infected by C. difficile or other pathogens that are very difficult to cure with antibiotics, but seem to do much better if you can reestablish a normal gut flora.
So C. diff is interesting, particularly with infants, because you actually see higher rates of colonization by C. diff in infants than in adults, but you almost never see illness from it. So C. diff in this population is really a weird one. And I look at that and I go, I don't know that we understand the whole community enough that I'd want to transplant someone else's random bacteria into someone else's kid, especially since infants will actually have a higher level of antimicrobial resistance genes than adults. So I kind of look at that combination and I go, oh, that's potentially very dangerous.
It was interesting that some years ago, many years ago, when I was an intern in pediatrics, and we were dealing with young children who had been treated with heavy antibiotic therapy and whose GI tracts were essentially, we thought, sterilized as a consequence. One of the recommended courses of treatment was to give them Lactonex, which was a probiotic with lactobacilli in it, which actually seemed to work. I was pretty impressed by that.
Yeah, it's especially, actually, a lot of the probiotics they give in infants are, they just split up the genus, so now a lot of them are Lactobacilli. It used to be Lactobacilli. Nothing can stay the same names this year. But so actually for my probiotic trial, I'm using an L. reuteri as the one I don't think will transfer, because I know that helps infants too.
Well, no other questions here. Do we have any questions online? I don't see any evidence for questions online. If not, thank you very much for stimulating talk, Diane.
Thank you.
Okay.
And if anyone has a grandbaby, I have study flyers.